Okay, so today's lecture we're looking at gluconeogenesis. Last time we discussed the citric acid cycle, also known as the Krebs cycle. So today we are looking at gluconeogenesis. Let's start with the definition of gluconeogenesis. So the word the word gluconeogenesis is made up of three words. Gluco to mean glucose, neo means new, and genesis means synthesis. So basically, this is a process um, of forming glucose from non-carbohydrate compounds. So basically, it is the synthesis of glucose from non-carbohydrate intermediates or precursors. So you can say precursors or compounds. So what are the major substrates for gluconeogenesis? So we have lactate, pyruvate, and also gluconeogenic, um, that is glucogenic amino acids, propionate, and glycerol. So these are the major substrates or precursors for the gluconeogenic pathway. The other thing to look for when we talk about gluconeogenesis is the location where this process takes place. So in terms of location, gluconeogenesis mainly occurs in the cytosol of the liver cells and the kidney. But the liver is the main site for gluconeogenesis, and it produces about 1 kg of glucose daily. So, in terms of location, it occurs in the cytosol of the hepatocytes and also the kidney cells, with the liver being the main um, gluconeogenic organ. What then is the importance of gluconeogenesis? It's important to remember that glucose is the main fuel in cells, and therefore gluconeogenesis aims to meet the needs. So it aims to meet the needs of the body for glucose when carbohydrate levels are not sufficient or carbohydrates are not available. So it's a source of glucose in the absence of sufficient carbohydrate amounts. A supply of glucose is necessary because the brain and also the red blood cells these are highly dependent on glucose as the primary fuel. So it's very important for the cell to get, the glucose because the brain and also the red blood cells, which are deficient, of the mitochondria, can be able to, access the glucose and produce energy via glycolysis. Also, gluconeogenesis aims to maintain levels of citric acid cycle intermediates. These are intermediates in the Krebs cycle. It also is important in clearing lactate, which builds up from skeletal muscle, active skeletal muscle, and also the red blood cells. It also is important or vital to clear a glycerol that is produced from adipolysis in the adipose tissue. It's also important because during times of fasting for more than a day, then the process of gluconeogenesis basically meets the basal requirements of the body for glucose. Very, very important. So we are saying in terms of the role of gluconeogenesis, it maintains the levels of the citric acid cycle intermediates so that the Krebs cycle continues to produce reducing equivalents which are going to be used in the electron transport chain. Also, it helps to clear lactate. So the end product of glycolysis, which is pyruvate, is converted to lactate in anaerobic conditions in the absence of oxygen, and therefore the product is lactate. Now, the lactate, when it accumulates in the muscles, it can become toxic, and therefore the lactate permeates the skeletal muscle cells into the blood, where it's taken to the liver, and then in the liver is then converted, back to glucose via gluconeogenesis. Also, the process helps to clear glycerol, which is produced from a breakdown of triacyl glycerols, which are the fats. So when you break down a triacyl glycerol, you have a glycerol molecule plus 3 fatty acids. So the glycerol that remains can then be converted to as we shall see later on, to dihydroxyacetone phosphate, and then utilized to re-synthesize the glucose. And then also in the fasting state, where somebody is not taking, carbohydrates, or they are not having a meal, the gluconeogenetic pathway is activated to meet the basal requirements, of the body for glucose. So those are the important aspects of gluconeogenesis. Let's look at the reactions of gluconeogenesis. So the reactions of gluconeogenesis actually closely resemble the reactions of glycolysis, except that it's the reversal. So we are saying these closely resemble the reversed pathway of glycolysis. However, it is not a complete reversal. This is because certain steps in the glycolytic pathway are not reversible. So 3 out of the 10 reactions of glycolysis are irreversible. So it is these 3 reactions which provide, or rather which make gluconeogenesis not to be a reversal of glycolysis. So these are thermodynamically unfavorable reactions. So to be able to make them work, then we need another enzyme to act other than the ones that we are using under glycolysis. So we are going to look at, the reactions under glycolysis, and then look at the reactions under gluconeogenesis as they occur. But it's important to take note that the 3 irreversible reactions are catalyzed by the following enzymes. So every time you've got an irreversible reaction in a biosynthetic pathway or in a pathway, just know that that's a point of regulation also. So the 3 enzymes, or rather the 3 steps that are irreversible, are catalyzed by the enzyme hexokinase. Number 2, phosphofructokinase. And the third one is the step catalyzed by pyruvate kinase. You will recall from glycolysis that hexokinase catalyzes the phosphorylation of glucose to form glucose 6-phosphate, which is the first step. Then you have phosphofructokinase, which catalyzes the third reaction, where you have phosphorylation of fructose 6 phosphate to form fructose 1,6 bisphosphate. Then pyruvate kinase catalyzes the last step in the glycolytic pathway, which is the conversion of phosphoenolpyruvate to form pyruvate. So those are the 3 regulatory states. Now, let's look at a complete set of these reactions. Now, we have said that gluconeogenesis is almost a reversal of glycolysis. So we'll start from the end of glycolysis and then begin to come back come downwards up to the point where we are going to form um, glucose. So the starting point really is to understand that the pyruvate that is formed, um, in the Krebs cycle goes into the mitochondria. So here we can say, first of all, the pyruvate can be converted to lactate via the enzyme lactate dehydrogenase through a reductive process. So you have NADH plus H plus, like that. So the pyruvate that is formed from the citric acid cycle, let me just change the color so that we can show the mitochondria. So the mitochondria is a double membrane structure. So I will not show the two membranes, but I'll just show that this is the mitochondria. So I'll try to make it a bit large, like that. So once the pyruvate goes into the mitochondria, it's converted to acetyl-CoA. So the acetyl-CoA then combines with oxaloacetate to form citrate, and then it would have entered, the citric acid cycle. So all these conversions, which I will not go through, because by now we did, the steps that are involved in the citric acid cycle. So you have fumarate, and then this is converted to malate, and then malate is then converted to oxaloacetate via a dehydrogenation process. Just add. So you have NAD plus being reduced to NADH plus H plus, like that. Okay, and the enzyme that is involved in the conversion of malate into oxaloacetate is malate dehydrogenase. So I'll abbreviate it as MDH. So it's malate dehydrogenase. Now, what is so interesting is that once pyruvate enters the citric acid cycle, it doesn't come out as pyruvate because it enters the citric acid cycle. So the pyruvate will leave the mitochondria because the mitochondria is not fully permeable to different substances. So it will leave the mitochondria as malate. So because there is a malate shuttle. So once malate is shuttled out of the mitochondria, then it is converted by the same enzyme within the mitochondria, which is malate dehydrogenase. So it's a replica of this step here in the mitochondria, where you have NAD plus being converted to NADH plus plus a hydrogen. So that is the reduced form of NAD plus. So we are forming here oxaloacetate. The oxaloacetate is then converted to phosphoenopyruvate via a catalyzed reaction by the enzyme phosphoeno. Let me put it in a different color. Via another enzyme which is phosphoenopyruvate, carboxykinase. So it converts the oxaloacetate into phosphoenopyruvate. Within this state, there is a utilization of one GTP molecule as a source of energy. So we form GDP. And then we also have said here carboxykinase. So it actually removes one carbon atom as a CO2. Okay, so the whole process here really is just to show how we are getting back, the carbon atoms that we are taken into the citric acid cycle. So once we convert the pyruvate into acetyl-CoA, the acetyl-CoA then enters into the mitochondria, and then in the mitochondria, the acetyl-CoA combines with oxaloacetate to produce citrate. Then citrate is converted to all these intermediates, which is alpha-ketoglutarate, to succinyl-CoA, fumarate, malate, and then back to oxaloacetate. But then in an event where we need to synthesize glucose from non-carbohydrate intermediates, then we're going to get malate, shuttle it out of the mitochondria, and then convert it back to oxaloacetate. Then oxaloacetate is converted to phosphoenopyruvate via the enzyme phosphoenopyruvate kinase. Now, once we get the phosphoenopyruvate kinase, you'll remember that the pyruvate and phosphoenopyruvate, these are steps in the glycolytic cycle. So that is step number 10 and step number 9. So let's go backwards and see how we are going to move into the glycolytic cycle, so that we see the opposite of, or rather the reverse of glycolysis. So we are on step number 9. So we are saying phosphoenopyruvate. So we have phosphoenopyruvate, which we have produced. So the phosphoenopyruvate is then converted via a reversible reaction to 2 phosphoglycerate. I think let me put this one on the side. Sorry about that. So let me put the phosphoenopyruvate on the side so that I can indicate the other process. So we have phosphoenopyruvate, which is converted to 2 phosphoglycerate, which is converted via a reversible reaction to 3 phosphoglycerate. The 3 phosphoglycerate is then converted to 1,3 bisphosphoglycerate, and then the 1,3 bisphosphoglycerate undergoes a dehydrogenation reaction to give us glycerol. Let me just glycerolaldehyde 3-phosphate. Let me just write it in full. So glycerolaldehyde 3-phosphate. So this reaction so you have NADH plus H plus to give us NAD plus like that. So the reverse requires an input of a reducing equivalent of NADH to produce the glycerolaldehyde 3-phosphate. On the other side, if you recall from glycolysis, we need dihydroxyacetone phosphate. We need dihydroxyacetone phosphate. So the dihydroxyacetone phosphate comes from glycerol. And I did mention earlier on that the glycerol comes from adipolysis or breakdown of triacyl glycerols, which give out fatty acids and also a glycerol molecule. So the glycerol once produced there is then phosphorylated by glycerol kinase. So we use one ATP molecule. So the enzyme here is glycerol kinase. So it phosphorylates a molecule of glycerol to activate it to glycerol 3-phosphate. So we are getting glycerol 3-phosphate. So the glycerol 3-phosphate is then acted upon by glycerol 3-phosphate dehydrogenase. So we are using NAD plus, and then we are getting a molecule of NADH plus plus. Let me just write that properly. So we are getting NADH plus plus hydrogen ion. The product of this is dihydroxyacetone phosphate. Which is abbreviated as DHAP, dihydroxyacetone phosphate. The enzyme here, if I can put it in a different color. So we said that is glycerol. So that is glycerol 3-phosphate dehydrogenase. Okay, so forgive me for that. Okay, so glycerol 3-phosphate dehydrogenase catalyzes the conversion of glycerol 3-phosphate to dihydroxyacetone phosphate. So this step now, once we have once we have our glyceroldehyde 3 phosphate and the dehydroxy acetone phosphate, the two combine to form fructose 1,6 bisphosphate. So we can go to the next step now. So where we're saying now, so we are saying glycerol 3 phosphate, glyceroldehyde 3 phosphate plus dehydroxy acetone phosphate. The two will combine to form fructose 1,6 bisphosphate. Fructose 1,6 bisphosphate. So the fructose 1,6 bisphosphate is then reduced. Now I'll put on one side I'll put glycolysis. On the other side I'm going to put gluconeogenesis so that we see how these move. So on my right hand side I'll put glycolysis. On this side we put gluconeogenesis. And I think let me put them the other way. Glycolysis, let me put it on my left hand side. So glycolysis, and then this side I put gluconeogenesis. Okay. So we are saying under gluconeogenesis, we are reducing the fructose 1,6 bisphosphate to fructose 6 phosphate. So to move from fructose 1,6 bisphosphate to fructose 6 phosphate, we are supposed to remove an inorganic phosphate molecule. So let me put that. So to move from fructose 1,6 bisphosphate, we are actually dephosphorylating the molecule. So here we are saying that we add water, we get an inorganic molecule. This side in glycolysis, we are saying we are adding a molecule of a phosphate molecule, and then we are getting out ADP. Now, in glycolysis, the enzyme that is involved for this conversion is fructose phosphofructokinase. So a kinase adds a phosphoryl group to move from fructose 1,6 bisphosphate to fructose 6 phosphate. We are using the enzyme fructose 1,6 bisphosphatase. Okay. So a phosphatase removes, it teases out, a phosphoryl group. Okay. From here, the next stage or the next step is to convert the fructose 1,6 phosphate into glucose 6 phosphate, or rather to convert the fructose 6 phosphate to glucose 6 phosphate. If you remember remember, in glycolysis, this reaction is actually just an isomerization reaction, and it's a reversible reaction. So we are interested in the reactions that are non-reversible. So the conversion of fructose 6-phosphate to glucose 6-phosphate is catalyzed by the same enzyme, the hexose isomerase enzyme. So it's the same one that we're going to use. But to convert the glucose 6-phosphate to glucose, or rather to the just normal glucose, we need another enzyme. Okay, so to convert from that to glucose. So in glycolysis, the enzyme that phosphorylates glucose to glucose 6-phosphate is hexokinase. So here we are adding a molecule of, or rather an inorganic phosphate group. So we are getting ADP. We are saying this is catalyzed by the enzyme hexokinase. For gluconeogenesis, to move from glucose 6-phosphate to glucose, again here we are removing a phosphate. So we are removing a phosphate. So we are getting out an inorganic phosphate molecule. The enzyme that is involved for this removal is glucose 6-phosphatase. So like I said earlier on, a phosphatase will remove a glucose, it will remove an inorganic phosphate, whilst a kinase will add a phosphoryl group. A phosphatase will remove a phosphoryl group or dephosphorylate. A kinase will phosphorylate or add an inorganic phosphate group. So the areas of concern that we have to be aware about is the first step, that is catalyzed by hexokinase, in which the reverse step is catalyzed by glucose 6-phosphatase. And also the third step, which is catalyzed by phosphofructokinase, in which case the reverse is catalyzed by fructose 1,6-bisphosphatase. And then also the other point of regulation, which we have mentioned, is the step catalyzed by pyruvate kinase, which is the 10th step here. So pyruvate kinase. So these are the control points which are different between glycolysis and gluconeogenesis. So gluconeogenesis consists of about 12 steps compared to the 10 steps of glycolysis. The reason is because the first steps involve the conversion of malate, the first two steps rather, malate into oxaloacetate, and then the oxaloacetate is converted to phosphoenopyruvate, and then the phosphoenopyruvate is converted to 2-phosphoglycerate, then 3-phosphoglycerate, and so on until we produce the glucose finally. So those are the steps for gluconeogenesis. We move on to regulation. Okay. So I think the other aspect that we should talk about here is, before I come to regulation, sorry, is that the process that we've defined so far, or described so far, comes from carbon units from a carbohydrate. So these are just recycled. But we can also have gluconeogenesis coming from amino acids. So we can have gluconeogenesis from, say, amino acids. So gluconeogenesis can occur from amino acids, from glucogenic amino acids, which result in the formation of pyruvate. We can also have gluconeogenesis from glycerol, as I have indicated in the pathway. This one, the glycerol originates from breakdown of triacyl glycerols or tags by lipolysis. This is later converted, as I showed earlier on, to dihydroxyacetone phosphate. We can also have gluconeogenesis coming from propionate. So propionate results from oxidation of odd chain fatty acids. As we will discuss later on, you will realize that the breakdown of fatty acids, most of the fatty acids are even numbered, but we have a few that are, have got an odd number of carbon atoms. So when they are oxidized, because the slicing of the carbon atoms is done um, every 2 carbons. Okay, so the last group of the carbons come out with 3 carbons, which is the propionate group, and this is what actually goes on to into gluconeogenesis also. So this can also be used. So propionate can also come from the breakdown of methionine, the amino acid methionine, and also isoleucine. So these also can result, when they are broken down, into propionate. The other source of the precursor is lactate. So lactate, we did mention that this comes from um, active muscles, and this is the major precursor for gluconeogenesis. So this is the major precursor for gluconeogenesis. We know that under anaerobic conditions, pyruvate is reduced to lactate by lactate dehydrogenase. And the lactate, once it's reduced to, so the pyruvate once it's reduced to lactate, lactate has nowhere to go. So, so the lactate dehydrogenase enzyme converts the pyruvate into lactate, and remember this is in the muscles, and from the muscles this is then able to permeate out of the muscle cells into the blood, and then it's taken to the liver where it's converted back to pyruvate. So the conversion of pyruvate to lactate and lactate back to pyruvate, and then pyruvate synthesized into glucose is referred to as the Coronary Cycle. So the Coronary Cycle, as a summary, we can show it this way. So you have a muscle cell. This is a bad muscle cell. So we have a muscle cell, which is an active muscle. And then this side we've got, the liver. So we're saying that is our muscle, and this is the liver. So what happens is that within the muscle, glucose is utilized. So we can, let me try and use another color. So within the muscle, the glucose is utilized through the glycolytic cycle, as we mentioned earlier on, to produce glucose 6-phosphate. The glucose 6-phosphate can be converted in the muscle if there is no need for energy to glycogen. Can be converted to glycogen. If there is need for energy, then it undergoes, it goes through the glycolytic cycle to produce pyruvate. The pyruvate, because of anaerobic conditions in the muscles, then produces lactate. Within the muscle, the pyruvate can also be converted to alanine by amination. The lactate can then permeate the muscle cells into the blood, then it's carried to the liver. So the lactate comes to the liver. Once in the liver, the lactate is converted to pyruvate. Okay. The alanine also can be transported in the blood to the liver. Once in the liver, the alanine can be deaminated to form pyruvate. The pyruvate can undergo gluconeogenesis to produce the glucose 6-phosphate, which can be converted via glycogenesis to form glycogen. If we are still short of glucose, then we produce the glucose in the liver, and then the glucose can then be transported to the muscle. So the Cori cycle is just these conversions of um, glucose in the muscles broken down finally to pyruvate. Then the pyruvate um, is converted to lactate, because in the liver, like we have said, in the muscles, like we have said, here we have low levels of oxygen, so we're going to end up producing lactate, and the lactate then is able to be transported in the blood to the liver. Then once in the liver, it's converted to pyruvate to glucose, and then the glucose again comes back to the muscle. So that is um, the Cori cycle. Okay, let's look at control of gluconeogenesis as we wind up. So the control of gluconeogenesis is actually reciprocally, you know, done. So what this means is that you have gluconeogenesis and glycolysis. So these are actually, um, controlled more or less in reverse. When one process is active, the other one is made to be inactive. So when gluconeogenesis levels or goes up, it means that glycolysis is inhibited, and vice versa. And the points of regulation are the enzymes that we've talked about. Okay, so the first enzyme that we talked about in the regulation of glucose um, is hexokinase, and also we talked about phosphofructokinase in glycolysis, and then we also have fructose 1,6 bisphosphate. So this side, if I can put glycolysis, and this side I put gluconeogenesis. So for glycolysis, the enzyme that is involved is phosphofructokinase, abbreviated as PFK. And then on the part of gluconeogenesis, we have fructose 1,6 bisphosphatase. Okay. So as you would have it, these enzymes are activated and inhibited by um, different, small molecules. So, for example, fructose phosphofructokinase is activated by fructose, so it's upregulated by fructose 2,6 bisphosphate, so it has a positive effect. Then you also have AMP, so it actually activates glycolysis by activating the enzyme phosphofructokinase. The same enzyme phosphofructokinase, I'll put in red, it's inhibited by ATP. It's also inhibited by citrate. It's also inhibited by hydrogen ions. For fructose 1,6 bisphosphatase, it's activated by citrate, but it's inhibited by fructose 2,6 bisphosphate and also AMP, adenosine monophosphate. So here, actually, one thing which you should take note is that, for example, the ratio of AMP and ATP is a signal for energy levels in a cell. If the level of AMP is very high, because remember, you can have AMP plus two phosphoryl groups, then you have ATP. So that is the reaction. So if you have a high level of AMP, it means that the level of ATP is very, very low. And therefore, we need to, activate glycolysis so that we can produce, or rather we can increase the level of ATP. But if we have got, a low level of AMP, then it means that the level of ATP is very high, and therefore we need to block gluconeogenesis so that we do not make more ATP. So that is how you can look at it. The other point of regulation is pyruvate kinase, like we said earlier on. So I'll write the enzyme here. So in glycolysis, um, we are saying we have pyruvate kinase. So write the enzyme here, pyruvate kinase. In gluconeogenesis, we have actually two enzymes that work, but I'll just put one of them, which is pyruvate carboxy kinase, or we just say carboxylase. So for um, pyruvate kinase is activated by fructose 1,6 bisphosphate. It's inhibited by ATP. It's also inhibited by alanine. Um, then pyruvate carboxylase is activated by acetyl-CoA, and then it's inhibited by ADP. So these are the points of regulation. So I'll leave you to check out the other modes of regulation so that I can talk about, a hormone control. So the other aspect of regulation of gluconeogenesis is actually hormonal. So the hormone control here is mainly due to the effect of insulin. So insulin rises following feeding and stimulates formation of the enzyme phosphofructokinase. And also pyruvate kinase, meaning that insulin activates glycolysis, or rather, it favors glycolysis. But glucagon rises during starvation. So the levels of glucagon will rise during starvation. Therefore, it will inhibit expression of the enzyme fractokinase 1 and also pyruvate kinase. So it inhibits their actions. But instead, it promotes the expression it promotes the expression of key gluconeogenic enzymes. Which ones are these key gluconeogenic enzymes? So we have one, phosphoenol pyruvate carboxy kinase, and also fructose 1,6 bisphosphate. So the regulation by hormones is to activate the key enzymes that are involved in the two processes, or um, block their actions. So when the um, when the cell is in a starved mode, it will activate gluconeogenesis, in which case the levels of glucagon will rise, and then it is going to inhibit the expression of phosphofructokinase 1 and also pyruvate kinase, but instead will actually trigger the expression or the formation, or rather the activation of the key enzyme, which is phosphoenol pyruvate carboxy kinase, to convert the malate, uh, you know, or rather to stimulate the conversion of malate into phosphoenolpyruvate via malate dehydrogenase, and then the oxaloacetate converted to phosphoenolpyruvate. So this is actually the end of gluconeogenesis and how it is regulated. Thank you so much. I hope you have enjoyed the video. Okay.